Good morning, everybody. Just have to show you. There, can you see him? There you go. Baticus Maximus. We got a new Wi-Fi router recently because of various internet issues, and I named the Wi-Fi network the Animal Madhouse because that seemed pretty fitting. Okay, so all right, what do you want? She's turning into Steve Jobs of dogs. See the high-necked black collar. It actually says "boo" on the back. It's a Halloween thing. Anyway, let's chat, get that up as well. All right, so what we have to do is basically go through translation. That's what we have to do over the next today and on Wednesday, and then the exam is on Friday. And so translation is complex, and there's a lot of moving parts, and students struggle with it, right? In many ways, it's relatively straightforward, but you got to bring all of those different bits together to be able to get to that point. And that's the challenge. So, a sneezing cat. Anyway, I've uploaded a new version of the PowerPoint. I think I sent an announcement around last week with this video in it. There's also another video buried down there somewhere, which is a particularly good watch, right? And so actually I might even just kind of run that through. Do I have that now? Go, what's this one? Yeah, still open. Dog chasing cat. Sorry. Pepper, leave her alone. God dang it. And so now we have, where can you see it? Can you see she there? Yeah, there she is. Cat as well. And dog. Who knows whatever she's doing. Oh! Get off. Well, puppy doesn't have any sense of shared space. Get off of that. Okay. All right, so this is a real neat video because it actually puts together a lot of different things, right? So the one I usually show is just transcription and translation. That's this one up here, but this one actually shows splicing too, which I think is super cool. So have a look through that as well. We're not going to go through it today because, pardon me, we've already covered that. All right. It's all slowed down, which is why the sound sounds a little bit silly. Where we're at. All right, so now we have the RNA is in the cytoplasm, right? So we're talking about eukaryotes here. The same principles apply to prokaryotes, but there are some fairly major differences for a bunch of different reasons. So this is about eukaryotes, right? So what you'll see here is the ribosome. There's two subunits, right? So this is a real big protein complex, riboprotein complex, right? So we've got ribosomal RNA and proteins, different subunits make one large piece called a large subunit, unimaginatively, and a whole bunch of other proteins and at least one RNA that makes a small subunit. right? So in both prokaryotes and eukaryotes, the small subunit binds to the RNA first, right? So they do kind of different jobs. So the small subunit is all about mRNA binding and what's called processivity, which is just a fancy word for moving along the RNA, right? The large ribosome or subunit equals peptide bond formation. And polypeptide extension, right? So this happens in a particular sequence, right? As always, there's beginning, middle, and end, right? And these things assemble in a very particular order. So that's what this is going to show you now. So this is the small subunit coming in, right? And I know that this is a eukaryotic one because it binds to the end, right? And so this is one of the differences, which we'll go into more detail in a little bit. Then once the small subunit assembles on the mRNA, then the large subunit comes in and forms the functional ribosome, which is what you see there. So as we've learned already, got mRNA, which is the information, ribosome, which is the machine that makes the peptide bonds, basically. We've got amino ACLT RNA synthetases, which we're not going to see here because they're doing their job elsewhere in the cytoplasm. And we have tRNAs, right? These transfer RNAs, or as I like to call them, translator RNAs, right? These are what are represented by these molecules. And so these enter the ribosome, drop off their amino acid, and leave. That's what you're going to see here. So they come in and they come out the other end as the mRNA moves through the ribosome. And when we look at that in detail, right, so we kind of go into the actual ribosome here. There we go. What you'll see is ribosomes with amino acids. That's what these little red bubbles are. And if you look super carefully, you'll see them on the end of these tRNAs. Come in to the ribosome. Again, this is the large subunit, the one on the top. Drop off their amino acid and then leave, right? So there are 3 different sites in the large subunit which are involved in each of those different processes. And for every one, you can kind of see them, not very well defined here. For every one of these tRNAs that comes in, the ribosome moves along 3 bases of RNA. So if, oops, type that better. If these are the letters of RNA, then codons, which are groups of 3 letters, are the words. And it's the words, essentially the codons, that are recognized very specifically by the tRNAs. And essentially that just continues along and the new protein gets extruded out of the ribosome, like so. And that's pretty much it. So there's an awful lot that happens after this, right? This is not the end. There's a huge amount of protein machinery, right, which is involved in folding, processing, exporting, you know, slicing and dicing and all that kind of stuff of the protein, right? So this is kind of the building blocks, essentially, right? This is the first stage of protein synthesis. It's not necessarily the last. So I recommend that you go through this video. It's really, really good for visualizing what's going on, right? And really, because this is such an abstract concept, visualizing is key, right? Being able to literally in your mind's eye, be able to recapitulate what you're seeing in this video, right? Because if you can do that, then it's pretty easy, right? Okay, so let's go back a wee bit. So we finished up kind of around here, right? Talking about protein structure, right? And protein structure, of which you're seeing a variety of, I guess, substructures, right? Secondary structure in scientific parlance, is determined by the properties of the amino acids that constitute that protein, right? And that is determined by the R groups. There we go. And also what amino acids you have and the order that they're in. So this is what a ribosome looks like. So kind of going through those different components, essentially, of translation. This is the small subunit down here. And as you can see, that's where the mRNA goes, right? So this is all involved in recognizing the RNA and moving along it, right? And so some of the energy in translation, or that's used in translation, is used to move the ribosome along the RNA, or the RNA through the ribosome, depending on your perspective. Now this one up here, oops, if I select the right thing, there you go. This one here, this is the large subunit. And really they're called that because of their size, right? You know, when they were isolated, there was a large protein and a small protein. So that's what they call them. And this consists of an exit tunnel. This is where the protein kind of gets extruded out. And 3 sites, right? And so in order of use, right, they read APE. In order of, I don't know why it's always presented this way around. Maybe because that's the way we read words ourselves. So we kind of just translate that into, molecular machinery. It reads EPA, right? And so one way of remember this, what was his name? Dang it, can't remember. It was a presidential hopeful in 2016. Texan guy, Rick Perry. There you go. One way of remembering it is that this is the name of the government agency that you wanted to get rid of, that you couldn't remember its name of. The EPA, Environmental Protection Agency, or APE, doesn't matter. Anyway, so the actual proper terms are given here, but the way I remember it, which is kind of easier, is A equals access, and that's the entry point, where the charged tRNA, and I mean charged as in it has an amino acid on it, enters the ribosome, right? P equals pairing or peptide bond formation. And that's where, I'd say there's a couple of things that happen here, but the main one is where the peptide bond between the existing, oops, if I can spell that right, polypeptide that's being made, and the incoming or new amino acid is formed. And there's a lot that goes on in that particular spot. And E is easy, and it's the same one as what's up here. That's the exit. And that's where the uncharged, so a tRNA that doesn't have an amino acid anymore, leaves the ribosome. So essentially there's this constant flow through of tRNAs that they come in, drop off their amino acid and leave. And they then go round back to the cytoplasm and get a new amino acid added to them. Right, so there's a couple of different components to that. And one is obviously the tRNA, right, which has this particular structure. And so this is another example of funky structures, right, of RNAs. Sorry, allergies are going crazy at the moment. Yay for Texas. And so the two key points of tRNAs are the anticodon. This is the bit of RNA that recognizes the codon in the mRNA, right? So the anticodon is the opposite of the codon. That's why it's called an anticodon. And on the other end, you've got a place to add an amino acid. And so the thing that does that is the amino acyl tRNA synthetases. And so translation is an energy intensive process, right? Without a shadow of a doubt. And one thing that I often ask students is, where is that energy used, right? Because this is an important part of organismal biology, right? So if an organism starts being nutrient limited or even starved, the first thing that stops is translation. So you stop making new proteins because translation is extraordinarily energy intensive. The second thing that happens is that proteins actually start getting broken down for energy. Once you've used up all your fat reserves, which, you know, if you're being starved, you probably don't have many, then you start breaking down proteins into amino acids to feed those amino acids into the, which was it, TCA cycle, right, to make ATP to survive. That's why when you look at people that are in, you know, war zones or famine areas, they are wasting away, right, because they're literally eating themselves to stay alive. So energy is important, right? That's what drives a huge amount of biology, essentially, at a vast range of scales as well. So anyway, essentially what happens first, we've got this protein, and it has this very particular shape, right? So this is a space filling model of a amino acyl tRNA synthetase. And they have a very specific shape because they have to recognize a very specific amino acid and put that amino acid on the correct tRNA, right? And they do so by using the anticodon, right? So essentially the enzyme recognizes the amino acid at one end and the anticodon at the other end. That's how this is so specific, right? That's why one tRNA equals one amino acid. So anyway, what happens first is amino acid comes in as well as a molecule of ATP, right? So this is where one ATP is used in the adding of an amino acid to a protein, right? So even before, you know, translation begins, energy is being spent to make that protein, right? Essentially, there's this pool of charged tRNAs floating around that are ready for ribosomes to use. Anyway, once that happens, essentially the ATP is hydrolyzed, where it becomes AMP, which is kind of odd, right? Usually it goes to ADP. And when that happens, tRNA is allowed to enter. And it enters in a very specific way, right? The whole kind of shape deal of proteins is very, very important here. And so that tRNA fits, you know, like hand and glove kind of deal, right? Or almost like they're spooning, right? It fits very, very specifically, right? So the anticodon down here, the amino acid up here. And when the tRNA comes in and it makes a good match, the AMP leaves and that bond, that covalent bond is formed between the tRNA and the amino acid. Now, when that happens, protein changes shape and the tRNA, the charged tRNA is now free to leave, essentially. Right, so all of these components are essential to translation. And understanding what they do and how they do it is really important to understanding the bigger picture as well. Does anybody have any questions so far? Really about anything? Hopefully it will make sense, but you're going to have to figure out whether it makes sense on your own as well, right? Okay. All right, so let's get into the details then. So as with most processes, as I said, there's a beginning, a middle, and an end. I'm going to go through prokaryotic translation first, because in some ways it's a little bit simpler. Okay, so essentially in prokaryotes, mRNA molecules are pretty simple. And literally as soon as the start codon, by the AUG down here, and this sequence called the Shine-Dalgarno sequence, named after the people that discovered it, emerges from the RNA polymerase, this mRNA can begin to be translated. Right, so as I've said earlier in this topic, in prokaryotes, transcription and translation can occur at the same time. In fact, they usually do, because they share the same space, and that's one of, you know, the ways in which bacteria are able to do what they do. So in bacteria, we have, same as in eukaryotes, a large and a small subunit. The actual sizes are different. The S stands for, I think, Stromberg units, which is basically where they end up after centrifugation in the column of sucrose. I think it also stands for sedimentation too. But anyway, relatively straightforward in bacteria. Essentially, we have the start codon, which is the beginning of the open reading frame. And just upstream of that, we have the Shine-Dalgarno sequence, right? So to allow that small subunit to bind, we have an IF. So IF equals initiation factor. And it's EIF in eukaryotes. That basically brings in the small subunit, right? And prevents the large subunit from binding, right? So the small subunit has to bind to the mRNA first, right? If it's bound to the large subunit, then this just doesn't work. right? It has to bind to the mRNA first. As soon as it does, right, and essentially this Shine-Dalgarno sequence is a sequence in the mRNA which is recognized by the small subunit and the IF3. And that directs the small subunit to that specific spot. And the aim of the game is to get the P site, this site here, the one in the middle. Hey love, dogs have been fed. One in the middle to be directly over the start codon. Right, so this spacing is super, super important because when the large subunit comes in, the P site is going to be bang over the AUG. So now we have a whole bunch of other stuff, right? We have some other initiation factors. We have our tRNA, right? So the very first tRNA is always methionine. Oh, no, I just fed the dogs before starting class. I got home a little bit earlier than I thought. Saved my wife the job. My dogs don't bark for food, they just pester. They just kind of like get underneath your feet and look at you sadly until you give them some food. Yeah, lucky in some ways, I guess. Where was I at? It's always met tRNA. So the first amino acid or peptide, that's probably a better word, in a protein is, at least as it's first synthesized, got to add that caveat there, is always methionine. Not necessarily in a mature protein because it depends on what happens to the protein after. So we have our charged FMET tRNA. Its anticodon is the opposite of AUG, right? So here comes that base pairing again, right? Which we've come across many, many times in different forms already. So the U base pairs with the A in the start codon, the A with the U, and the C with the G, right? So this FMET tRNA only recognizes an AUG, nothing else, right? It's very specific. And the other thing that comes in is GTP, right? So GTP is very similar to ATP. And essentially, when it gets converted to GDP, it releases energy, right? So here's another unit of energy that's used, right, to bring in the tRNA. So all of this jazz falls off, leaving behind just the tRNA. What's that dog doing? And then the large subunit can come in. Does that make sense? So small subunit binds, FMET tRNA binds to the start codon, which is in the P site, or will be. GTP is hydrolyzed to GDP. All this other stuff falls off, and that allows the large subunit to bind to the small subunit and form the functional ribosome. And in doing so, we now have our first amino acid in the P site, or the first charged tRNA in the P site. And there's a real neat video down here. Let's see if I can. Get that open. Okay, so this is for prokaryotes. It's really worth working through this because it does it in a very slow, deliberate way, right? And it also details the different energy used, right? So essentially, for every amino acid added to a polypeptide, you need 1 ATP for the, oh, I have to type this out, amino acyl tRNA synthetase, and 2 GTP. 1 to bring the tRNA in, and 1 to move the ribosome 1 codon along the mRNA. So this is the initiation that we're just talking about. You can see that we've got the FMET tRNA in the P site. Now we are able to have the next tRNA come in to the A site, right? And what that does is it puts 2 amino acids close together. So let's scan along a little bit. Okay, so here's the next one. This is a tRNA for serine. Again, you can see that the anticodon, AGG, is the opposite of the codon that's going to be red, which is UCC, right? So again, we've got our elongation factor now. So EF equals elongation factor. Right, because we've already initiated translation, already started, right? So that brings in the tRNA along with some GTP, like so. Now we need to do 2 things. To add an amino acid, we need to do 2 things. 1, we have to remove the amino acid from this tRNA, right? Because we can't have tRNAs dangling off of our protein, right? We've got to chop them off, done their job, off you go. Right. So we need to remove the amino acid from the first tRNA. And that's going to be methionine in this case. Hey, stop that. And then the second thing is need to form a peptide bond between the newly released amino acid and the incoming amino acid. Oh, hello, girl. Oh, there's Luna. Sentinel dog. World's worst guard dog, aren't you? She likes people too much. Aww. Sorry, getting distracted. Right. And that's what the ribosomal RNA in the large subunit does. So the protein that forms this structure doesn't do that job. It's the ribosomal RNA, which is in this region here. It's kind of like teardrop shape, kind of. region that lies between these two amino acids. Yeah. Let's see if I can do. All right, so there you go. This is our peptidase transferase, right? Highlighted in yellow just there. And so the first thing it does, I missed it, is it cuts off the amino acid from this tRNA, right? I don't know why it says Matt here. Maybe it knows that I'm watching the video. Isn't that spooky? And once this is free, then it forms the peptide bond with the next one, like so. Now we have an uncharged tRNA and a charged tRNA with 2 amino acids added to it. So what we do now is we have to move the whole process along, right? So essentially the ribosome moves along and the tRNAs stay where they're at. So that has the effect of changing where those tRNAs are relative to the ribosome. And we need a little bit more energy to do so. Just trying to find out where that is. Yeah, that's just in more detail. Here we go. Hang on. Oh, and the subtitles are off. Weird. Anyway. So you can see there's another elongation factor coming in. And it also has just, kind of missed it a little bit, unfortunately. There you go. It has GTP attached to it. Hydrolyzing of that GTP, right, releases energy that moves the whole ribosome along one codon. So 3 letters, which is what you'll see. It's just happening. It's happening. There we go. Just moved along, right? So the ribosome is processing along the mRNA, or depending on your perspective, it's pulling the mRNA through it, right? Like sailor climbing along a rope. Now we have the discharged or uncharged tRNA in the E site, free to leave, right? Has no other role in translation until it gets recharged back up here by the amino acyl tRNA synthetase. So it's like this constant cycle, right? Kind of comes into the ribosome, drops off its amino acid, pops back out, gets recharged, goes back in again as needed, essentially. Right. Now we have, let's see if it's a little bit clearer. There we go. Now we have our new, or what was the new tRNA with 2 amino acids, the one that was there before it and the one that it brought in the P site. The A site is now open. So now we can do the same thing again. Let's bring it back over here and bring in the next tRNA, right? Same thing happens again. Elongation factor and GTP bring the tRNA in, which is what you see just like so. Then these 2 amino acids are removed, right? So the bond is broken here between the tRNA and the serine, and then a peptide bond will be formed between the serine and the lysine. So now you can also see, which is really neat about this video, is how these words in the mRNA equals amino acids in the polypeptide. Because the order of these words and what they spell, you know, in this case, this is going to be methionine, serine, lysine. I don't know what GCC encodes. I'd have to look at a codon table, right? That now spells the order or the identity and the order of these amino acids. Right, so here is literally the translation of the information in mRNA via the anticodons, right, the specificity of those tRNAs and the amino acids that they bring in into a sequence of amino acids. And then the same thing happens again. Now we have 3 amino acids, so this is a tripeptide. And the ribosome translocates along. The uncharged tRNA leaves, so this was the one that brought in serine, right? It's no longer needed. And the A site is now open for another tRNA to come in. And this just happens over and over and over and over again until translation is terminated. Any questions? There's a lot to take in. Wonder if my puppy needs to go out. Yeah, so this, Bianca, is something that is worth going over several times on your own, going through the videos, going through the PowerPoints, and just kind of drawing out, right? So usually what, you know, this is the downside of teaching online, or one of them at least. Usually I just draw this on the board, right? And I'd recommend doing the same because that kind of muscle memory, in a sense, right, of just drawing stuff out will help turn this kind of, you know, 2-dimensional on-screen form of information into something that you're creating, right? And so it's going to stick a little bit better as a result. And in terms of elongation, translation is pretty much the same between eukaryotes and prokaryotes. The initiation, however, is different. Let's go back here. So this is, so essentially in prokaryotes, the whole thing assembles, the whole thing being the ribosome, around the start codon, right? With the aid of the Shine-Dalgarno sequence, right? Kind of pretty much assembles on the start codon and starts from there. In eukaryotes, however, it's different, right? So remember with eukaryotes, we have this untranslated region on either side of the open reading frame or the codon sequence. And remember, I've been kind of leading like the hints that these 2 regions are very important in translation, especially initiation of translation. And so that's because the cap is where the small subunit assembles. I'm going to show that in a little bit more detail over here. Right. And so essentially there's a whole bunch of eukaryotic initiation factors. Remember the EIFs? They assemble on the 5' cap because it has that weird ass structure, right? That doesn't look like RNA. That's the whole point of protecting the RNA from RNases, at least at that end. And because it looks weird, it can be recognized as such. And that's what these initiation factors do. And so now we have a whole bunch of other initiation factors, and there's a lot of them. Those bring in the small subunit. Notice it's a little bit bigger here. There's 30 proteins about in this. Imagine that, 30. That's kind of crazy. And it brings in the FMET tRNA, right? So our initiated tRNA. And the end result, essentially, we have a small subunit. We have a bunch of initiation factors, right? And their role is also to stop the large subunit from binding when it shouldn't. And all of that is, and our FMET tRNA, all of that is assembled on the 5' cap, right? And that often actually is with the aid of the poly-A binding proteins, which is kind of cool. If you remember talking about how they protect the 3' end from degradation, they also kind of loop back and interact with the stuff at the 5' end. So now, rather than kind of assembling on the start codon in eukaryotes, you assemble like some way upstream, right, in the 5' UTR, and the small subunit scans along the mRNA until it finds a start codon, until it finds an AUG. And you can bet your bottom dollar there is no AUG, no other AUG in that 5' UTR. No worries, Isaiah. Catch up with anything that you might miss in the next few minutes through the video, okay? Hope everything's okay. So we now have this scanning, and basically what it uses, it's like a barcode scanner almost. It uses the anticodon of the FMET tRNA to find the start codon. And it goes, nope, 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 nope. Ooh, match, right? Binds by base pair complementarity, right? So even if it finds an AU, that's not good enough. It has to be an AUG for it to stop and go, and the start codon. Once it does, a whole bunch of other stuff happens, right? Essentially, all of the initiation factors hoof it, fall off, and the large subunit, which is even bigger, hence it's called the large subunit, with a bunch of ribosomal RNAs and a ridiculous amount of proteins, oops, now assembles on the small subunit. But the same kind of idea applies, right? Now we have our FMET tRNA in the P site, and our A site is open. That's exactly the same as what we have here in prokaryotic translation, right? So you see this, it's like the end is the same, the means of getting there is a little bit different, right? And as soon as that happens, now we have our next one. This is phenylalanine, right? So the tRNA for that recognizes the UUU. We use still GTP to bring that into the A site. Peptide bond is formed. Another GTP and an elongation factor comes along, moves along one base, whole thing starts all over again. Right, and so as with transcription, the actual bit in between, right, the middle part, the elongation part, is pretty routine, right? Once it gets going, and the initiation is the complex bit, it just continues until it's told not to, right? And we'll cover termination on Wednesday, and then we'll go through some of the quirks, some of the differences between eukaryotes and prokaryotes, and a little bit about degeneracy and wobble, right? And that's it for me today. So again, lots of stuff to take in. It's really worth using those videos and trying to kind of match stuff up in your head, right? How does the stuff that you see in PowerPoint tie into the video, and how does that all kind of make sense in your head, right? Awesome. Well, have a lovely day. It's a bit grey, but you know, take it as you find it. And I will see you either tomorrow or on Wednesday. Have a good day, everybody. So Bianca, I'll send you an email and we can work around that. So don't worry if you are dragged into that. We'll just, you know, figure out a way of accommodating that. Okay. So don't stress about that, but I'll send you an email anyway. Okay.